or any UK staff over there if it's not safe to do so. And I think we managed it. There was some heavy notes.
Take a couple of questions from the audience. Any questions for me? Or move on? No? Well, thank you again, Neil. Very, very interesting. So moving on to our second talk of the afternoon, and this is from Professor Mark Allen, who's from the University of Warwick, and he's going to enlighten us on clinical application of microbial genomics and metagenomics. The future is here.
Well, great. Thank you very much for the invitation to speak. It's going to be hard to follow that last act, I think, and particularly as it's the middle of the afternoon, you're going to all start falling asleep, but I'll try and keep you awake. What I'm doing also is I'm recording this, so if any of you do fall asleep, I shall put it up on YouTube and you can watch it tonight. And anyone who you know wants to watch it can watch it later. So what I'm going to do is very, very quickly take you through some bacterial pathogenomics and high-throughput sequencing, just an overview of that, and then take you around the world. So we're going to go to Birmingham, Germany, then we're going to go to West Africa, quite topical given the last talk, and then come back to Birmingham, and then we'll talk about the cloud. So I was very lucky to live through this great revolution in microbiology around the turn of the millennium when we got the first glimpses of bacterial genome sequences. This gave us the first glimpses of the inner workings of humankind's most fearful adversaries, most fearful microbial adversaries. Things like the causative organism of the Black Death or of the White Plague, tuberculosis. And when I give a rather longer lecture, I put this slide in and actually wax lyrical, quoting some poetry, because there's a quote there from Keats. And it really was very much like that Voyager program where probes went through the solar system and turned smudges down a telescope into newly mapped worlds. For the first time, we really could get a detailed view of the inner workings of these bacteria. Then you fast forward over time, over about a 10-year period, and something changes. So when we started off around the turn of the millennium, sequencing was still very expensive. And it was like an Apollo mission. It was not something the average lab could ever think of doing. But sequencing got better and better in terms of performance, price, user-friendliness, and so forth, following Moore's law for a few years. And then suddenly, around 2007, the foot went on the accelerator, and it went into what we might call hyper-Moore's law. And this was the arrival of what we often call next-generation sequencing or high-throughput sequencing. And this gave us the ability to do sequencing on a scale never seen before. So in the sense of you could do it locally, because you could do it one of these benchtop sequencers like a MySeq, and you could do it as a distributed activity. You didn't need to just go to the Sanger Institute and do sequencing. It could happen in labs around the world and around the country. Now, one interesting problem that we started to look at was this particular bacterium, Acinetobacter baumannii. And we had just bought our own MySeq in Birmingham, and we were faced with an outbreak with this organism. So this organism, I'm sure it's familiar to almost everyone in the room, gram-negative bacterium, multi-drug resistant, marching towards pan-drug resistance, causes problems in wounds, in bloodstream infections. During the Afghan and Iraq conflicts, it was often seen in military personnel returning to the UK with transmission from them onwards in hospital environments onto civilians. And this wasn't just in the UK, similar picture was seen also in North America and in parts of Europe as well. It presents some problems in the lab. It's quite difficult, or it's pretty much impossible to distinguish between different species using phenotypic tests. And the other problem is that often in hospital environments you will see serial clonal outbreaks. So the organism will come into the hospital, sputter on for a while, then disappear. Maybe you'll have a bit of a time without much Acinetobacter, then another strain will come in. But the problem is that although you can, even using methods that have been around for some time, identify a new outbreak and say, oh yes, you've got a different sequence type in your lab, in your hospital, you haven't up until recently been able to glimpse deep inside the outbreak and look at the mode of spread and the transmission chains within the outbreak. And that's what we were hoping to do with next generation sequencing. So the outbreak that we were investigating ran from 2011 to 12. It was the first case reported in a military patient, and the Queen Elizabeth Hospital, Birmingham, was, and still is, in fact, the first port of, called the first destination for repatriated military casualties. So this patient brought the Acinetobacter in. It was sent to Colindale, and they said, oh, this is pulsotype 27. You've never seen this before. This is a new outbreak for you. And then the outbreak went on and on and on and lasted for 80 weeks and involved 60 patients. So a very substantial outbreak. I'm not aware whether it's the largest outbreak or the longest outbreaks ever seen in this country, but it must be quite close to that. So during the course of the outbreak, we genome sequenced 91 isolates from patients and from the environment on the MySeq. And we were able to confirm when we did the first index strain genome that it was different from all other strains that have been sequenced, where there are sequences available in the public domain, and it was different from anything we'd seen locally. Now, moving very quickly through this, what we did was we sequenced all those genomes. We called out the variation within those genomes. So we started off with what the index case over here, which we called genotype 1, and then we called out just single base changes, single nucleotide variants, just simple spelling mistakes in the genomes as time progressed. And each of these red numbers here is a different SNP that has occurred in the genome. And we were able to group the isolates we saw into a series of major genotypes. But what we also saw was there was this kind of spoke and wheel and spokes type of arrangement where you'd have a major genotype, then you'd have a couple of other isolates or a handful of isolates that had just one SNP different. And we were very pleased that we could actually make sense of this outbreak, because when we started this, we didn't know if the data would actually make any sense at all. But it was nice to see that there was a progressive accumulation of SNPs as the outbreak went on. So when we went from genotype 1 to genotype 2, there was this SNP number 2, but then all subsequent isolates had that SNP. When we went from genotype 2 to genotype 4, we had 5 and 9, and then everything else had that. So it was gratifying that we could actually make sense of the results and put these things into a nice, tidy kind of order. But things actually get more even clearer when you overlay that information about the genotypes on traditional shoe leather epidemiology, about the time and place where the patients were and when. And so this here we've color-coded the various wards that the patients were on, and we can see that generally they're on the same ward at the same time, and that doesn't require an awful lot of explanation as to how the outbreak is spreading if patients are fairly close to each other. What you can see, though, is that it started off in this ward, what we call Ward 1 here, but around halfway through, it suddenly switched, and then it became predominantly a problem on the burns unit. And that was harder to understand because the burns unit patients were all isolated in single rooms with ensuite facilities and everything, and it was very hard to work out how on earth can this organism be jumping around in the burns unit like that. But we had, from this, we had salient information that it really was the same strain, the same genotype moving around between these patients. And so eventually the penny dropped. The common factor was that they were all going to the same operating theatre to have their burns debrided. And then when we went into the operating theatre, we could get isolates from the environment there. We genome sequenced some of those and showed that they also were very closely related, and that was the source. And so after a deep clean, the outbreak was initially controlled. It then sputtered back into life because someone got nursed on a bed they shouldn't have got nursed on. And then it went back into the burns theatre for a while, but then the final deep clean, and we got rid of it. So it was interesting what we used genomics. Now you could say, well, it's just an outbreak you've described. What was the point of genomics? But it was very helpful doing this. We could exclude isolates from the outbreak. So we did see among some of the patients Acinetobacter pittii, a completely different species, and we could identify that through genome sequencing. We could link all the patients together via their SNP genotypes and link them to the environmental isolate. So it provided a very powerful impetus to say, yes, this is happening in front of you. And we show, as I mentioned, we got these kind of spoken wheels with the genotypes, but what we also saw was sometimes in patients we could get a majority genotype and then a couple of variants within them. We also saw that some patients had mixtures. So they had mixtures of two completely different genotypes, or two completely different strains within Acinetobacter baumannii, or two different species even. And this, is a problem because it means that when you're picking a single colony and say, oh, I'll just take one morphotype and move on with that, you may be missing something. The other thing was that initially we started off doing this in batches. We were just gearing up to get started. As we got towards the second half of the outbreak, we were able to go from a colony to a SNP genotype in less than 1 week. So we were able to effectively equal the kind of service we would be getting if we sent stuff away and then waited for the result from the reference lab. Anyway, that was all written up in a paper a couple of years ago now in Genome Medicine, so if you're interested to see more of the details of that, that was an interesting proof of principle case. Around the same time, there was another much larger community outbreak that you've probably heard of this E. coli 0104H4. Some wag named it the sproutbreak because it was supposedly associated with bean sprouts. And we did a study on this. We got involved with some German collaborators, and we came up with what we call open source genomic analysis of this particular outbreak strain genome. Now, I'm not going to say much about that because it's now getting to be a fairly old story, but I'm going to use it as a link to the next part of my talk. So the funny thing was, when we did that study, we didn't actually meet the German collaborators who were co-authors of our paper in the New England Journal until after the paper had come out. And so we went over, so myself and Nick Lohman, who was working with me, to go and see these guys, Martin Effelbacher standing with me there, drinking champagne. And we said, where do we go from here? People will say, well, you got very lucky to get a paper in New England Journal, but you're just one-trick ponies, one-hit wonders. We said, how can we get around that? What are our unique selling points? I said, well, we know quite a lot about genomics and bioinformatics. What's your unique selling point? And Martin said, well, actually, he's got a freezer full of shit. He's got 200 fecal samples from the outbreak. I said, okay, I can feel a title of a rap song coming on here. Let's just sequence the shit, to put it in the vernacular. Okay, where was I coming from? Well, the other issue is, of course, diagnostic bacteriology is still stuck in the, you mentioned the 1920s. Diagnostic bacteriology is stuck in the 1920s. Diagnostic bacteriology is stuck in the 19th century because the techniques that we rely on were invented in the 1880s. Like Graham with his Gram stain, and Robert Koch was the first person to show that you could propagate bacteria on solid media and pick single colonies. Can't we do better than that in the 21st century? So what I said was, why don't we just sequence those fecal samples and see if we can actually get information about the outbreak strain directly from the feces without having to culture it. So instead of up the top there, which is what they've done before, where you grow it in pure culture and then get a sequence, can't we go directly to the sequence? So what you're doing here is you've got a whole mixture of different bacteria. You just analyze the cells, extract the DNA from all of them in one go, and then afterwards, in the bioinformatics stage, after you've sequenced them all, you then start to do all the heavy lifting of sorting them out and working out what's there. Now, I was hoping that we might just get a few sequences from E. coli, that, or, from the sugar toxin, that we could say, oh yeah, definitely we can detect it. But in fact, it turned out to be much more successful than we were expecting. The very first sample we looked at, when we aligned the reads from the stool metagenome against the reference strain genome, we saw about 20-fold coverage, which is not far off what you get if you were just sequencing a pure culture of the organism. And it was clear that our expectation, which is that E. coli is not very abundant in stools, is simply not true. In the disease state, it does bloom up and takes over that microbiome. And we were even able to call out the MLST type directly from the stool sample when we looked at this. We were trying to write this up for JAMA, for the Journal of American Medical Association, and we passed it past the editor, and he said, he sought expert advice, and I know his expert actually was David Relman, who said, it's a bit like cheating though, isn't it? Because you just aligned, you have that reference genome from the outbreak that you sequenced earlier, and you've aligned against that. But at the time that the outbreak was going on, nobody knew what was there. What you should be setting yourself the challenge is of reconstructing what was going on, getting that genome out without any reference. So it's a bit like, you know, if you're trying to do a jigsaw puzzle, it's a much harder ask to solve the jigsaw puzzle when you don't have the lid of the box to look at and work out what everything is. And so could we get the outbreak strain genome without knowing what it looked like in advance? And Nick Lohmann, who did this work, a very bright guy, very versatile intellect, he just thought, what am I going to do here? And he first sketched out the problem. So this is the kind of total coverage of reads, and this is the GC content. It's a way of smearing out all the different taxonomic variety that's in this sample. And he'd also taken all the sequences and just done a quick pass taxonomic annotation on them. And you can see that when you look at sequences that are present in more than 2 outbreak samples, you've just got this taxonomic zoo. And the idea that you could ever work out that there was a single organism causing an outbreak looks a pretty tall order. But he scratched his head and he came up with a simplifying assumption. He said, well, if this is really an outbreak, and we had, I think, 40-odd stool samples, he said, we should expect to see representation of the organism in at least half of our samples. So let's just subtract away all of the reads that are not present in at least half the samples. And that did lead to a great simplification of the taxonomy, but not quite enough to actually give us a single genome. But you can see that the pinkish stuff there, the Enterobacteriales, which the E. coli belongs to, are starting to look a bit more prominent. So then he thought, well, how else could he get to this? And then he came up with yet another brainwave, which was if he could subtract everything that was seen in the normal stool microbiome, that might help him. Now, luckily, he was a bioinformatician working in the middle of the night. He didn't have to suddenly jump on a plane, fly to Germany and get 40 Germans off the street and say, give me your stool sample, because there were ongoing stool sample collections and metagenomic sequencing going on under a project called Meta and the Human Microbiome Project. So he just took the data that was available online and matched it for age and sex for these samples and subtracted everything that was in those and ended up pretty much with a pure genome where most of the stuff, all the stuff that could be annotated and could be assigned belonged to the Enterobacteriales. We now know that the other stuff is actually stuff kit contamination. And then now if you're still awake this afternoon, you will have said, well, hang on, if you go and subtract everything that's in normal people, you're subtracting 80% plus of the E. coli genome because we're all carrying the core genome of E. coli. So the final step he had to do was actually fish that back out again. I won't go into the details, but he ended up reconstructing the outbreak strain genome and getting reasonably good coverage of it from these samples alone. So that was an interesting exercise, just a proof of principle yes, you can make diagnoses just by sequencing the samples directly. Nick went on after he left my group. Well, he stayed in Birmingham, I moved to Warwick, and I just give a shout out for this, given this earlier talk. Nick went on and did his, I guess the people at PHE will say these were the cowboys. The cowboys went out there and they sequenced using another sequencing platform called Oxford Nanopore, and were able to get sequences of Ebola virus done in real time. But anyway, that's his story, not mine. But it's interesting if you read the paper. What I went on and did was we sequenced genomes from 200-year-old mummies from Hungary, and we got TB genomes out of these mummies. And again, as time is short, I can't give you, I'm not going to give you a lot of details about this. And in fact, if you're interested, the ASM made a lovely little app that you can download. I'll put the URL up there if you want to go and see more about this. But the thing was, we felt really cool that we'd got genomes out of TB genomes out of 200-year-old mummies. And in fact, my children were pretty impressed because Bill Gates retweeted my paper. And Bill Gates recognised what Dad's done. Wow, that's amazing. But of course, all of you in this room will say, well, you bloody academic. You know, what's the point in sequencing 200-year-old? You know, the good news is we've got a diagnosis. Bad news is it's 200 years too late. And so we would say, well, actually, nobody's done this on contemporary TB samples. Why don't we do that? And there's a phrase that's sometimes put over anatomy rooms, which in Latin there, which translates means the dead will teach the living. We thought, yeah, could we move on from this? And so we did. Going back to the future, we then undertook a study looking for metagenomic detection of TB in sputum. This was done by Emma Doughty, my PhD student shown there. And we published this in the journal PeerJ. Now, what she did was she extracted DNA from sputum samples using a variety of different protocols, and she got various, quite variable results. So in some of the samples, we got almost all of this DNA she was getting was human. In others, a larger fraction of it was coming from bacteria and from TB. And then when we mapped the reads to the TB reference genome, we got variable depth of coverage. In none of these cases did we get anything like full genome coverage, one-fold coverage, but we were getting, in terms of numbers of reads aligning, hundreds of thousands, tens of thousands, hundreds of thousands, in one case over 3 million reads aligning, so that was giving us 2/3 of a genome coverage. So this was enough for us to be able to identify not just that TB was there, but what lineage the TB strains belonged to. So we could actually make, on the basis of the information we were pulling out, a phylogenetic assignment, a typing assignment directly from the sputum. What we weren't able to do, is because we didn't get enough depth of coverage, this was the very first proof of principle study, was call out any SNPs that might be related to resistance. And so we have to be careful not to exaggerate what we've achieved there. But it's at least a first step. I'm going to give you one final little vignette before I finish. So in recent years, I've been interested in patients in intensive care unit. And many of you probably looked after samples from these patients. And you will know that when they come into the intensive care unit, particularly if they're a trauma patient, they have a healthy microbiome, they soon develop a sick gut microbiome because of all those antibiotics they get given. And this then allows them to get colonized with these highly adapted bacteria that live in hospitals and multi-drug resistant pathogens. And after colonization, these organisms, because there's no colonization resistance left in the gut microbiota, they can proliferate. So I was interested to sort of look at this in more detail, and we've just recently taken 10 samples from the intensive care unit and analysed them by metagenomics. This is not projecting particularly well. I apologise for that, I tried to put too much on it. We only did this work a couple of weeks ago, but one of the interesting findings is where we've actually tried to look at the phylogenetic assignment of the majority of the reads. And in some cases, well, in 3 of these cases, we've got Enterococcus faecium coming up as a predominant organism in the stool sample. So in one case, half of the reads that we were getting out of the stool sample from the entire ICU patient was coming from Enterococcus faecium. And I've run these through a program called Nullarbor, which is supposed to work on genomes, but in fact, there was such an abundance of these organisms that we could even call out MLST sequence types for the Enterococcus faecium and start to recognize some of the resistomes. Say, yes, this is not just Enterococcus faecium, it's vancomycin-resistant Enterococcus faecium, directly from sequencing stool samples. And more recently been looking at MLST results on all of the samples, and we've been able to identify Candida in some of them as well, and Stenotrophomonas. So in conclusion, it's clear when we start sequencing these stool samples that those microbiota in intensive care unit patients are very sick. They are very simple. They only have a few organisms in, not the hundreds you see in normal people. Pathogens are blooming. And in this setting, there's some preliminary evidence, at least, that the diagnostic metagenomics might be working. So where's all this going? I think that one of the interesting developments is that people are talking about sequencing DNA as a diagnostic approach, not just in microbiology, but in many other fields. And you may have heard of this idea of the liquid biopsy, where you can take blood from, say, a pregnant woman, and you can find out information about the fetus. You can take blood from someone who's had a transplant and work out whether the transplant's been rejected, and you can detect the transplant genome in the blood. And similarly with cancer, you can look at the cancer. So it may be that we're moving to something we might call macromolecular monitoring, where we start looking at all of these macromolecules in samples, and microbiology joins up with these other disciplines in doing that. One of the things, I just have to say this for a moment, one of the problems is, of course, analysing all this data, and I'm the director of a project called the Cloud Infrastructure of Microbial Genomics, Bioinformatics, or CLIME, and what we're doing is actually providing resources for people to do this kind of analysis. So if you're interested in doing bioinformatics, let us know, and we can provide you with some of the resources that we have available. So in conclusion, I don't think I put that bit about the future is here in the title. I think that was given to me when I volunteered to come here. But if we're looking at the future, here's a few quotes. So the first time I presented all that stuff about the German outbreak, I was actually in Berlin, not far from the Berlin Wall, where it had been. And I put this quote in from Lincoln Steffens as a bit of provocation, because that quote there, I've seen the future and it works, was made in the 1920s. He was an American journalist, and he went to the Soviet Union. And he went back home and said, oh, I've seen the future and it works. Obviously, the Soviet Union did work for a few more decades, but then eventually it did collapse a few decades later. Perhaps a more insightful quote there is from the author, the cyberpunk author, William Gibson. It says, the future's already here, but it's not very evenly distributed. And this is the problem. And I'm not going to stand in front of you and say that everyone's going to be doing diagnostic metagenomics tomorrow. What works in our setting when we're academics and we're highly skilled in bioinformatics may not actually translate elsewhere. And it's very difficult to see how the future, so that last quote there, prediction is difficult, especially about the future. Having said that, looking at the tremendous advances that we've seen in public health microbiology and its use of genomics through Public Health England and elsewhere in the recent decades, recent years, just in the last couple of years even, I don't think we can discount the fact that we may well be moving fast forward on all of these issues and even using metagenomics in the future. And that's me finished. And I'd like to acknowledge all the people who've helped me for this time. Thank you. Man over there, yeah.
Just wait for the program line, please.
Hi.
Hello.
What I wanted to find out in your experience is how much sort of budget and time do you require for one lab that doesn't even do any sequencing to establish a lab can manage the whole genomic sequencing in a MiSeq level?
At MiSeq level, it's about £70,000 to buy a MiSeq. And the various other bits and pieces have come to a few thousand more. And you've got to have a maintenance contract. I mean, I guess something like 100,000 is probably a reasonable sort of starting point in terms of budget. In terms of skills, I mean, the MiSeq is actually quite remarkable, we found, because unlike we used to have a 454 before the MiSeq, and we had one trusted individual who could run that, because it was so expensive and difficult and whatever that we wouldn't let anyone else loose on it. With the MiSeq, we've had at least a dozen people have done their own library preps and run them on the MiSeq, and we have PhD students using it, project students. I even had a gap year student come and make her own libraries and run them. She had a lot of help, but that gives you a measure of the fact that it is relatively easy to use as an instrument. So in terms of the getting started, I don't think it's a particularly difficult place to go. And we know that Public Health England is doing that. They are doing that centrally. The question is how far that should be sent out and distributed to other labs. The other issue is, of course, that technology is moving forward, and one of the annoying things is that they keep changing things. If you're wedded to standard operating procedures where you've got agreement from everyone around the table, this is how we're going to do it for the rest of time, and then they change the sequencing kits and they change all the procedures and all this kind of thing, it makes difficulties. So there are problems, it has to be said. The other thing is that nanopore is the one to watch. Nanopore has been kind of promised for quite some time. And I've been rather sceptical. They keep kind of promising us jam tomorrow, you know, like one of these cargo cults in the South Pacific that, you know, suddenly it's all going to come and it's going to change the world. But I think we are getting a glimpse that they are going to get a serious technology out there soon. So, yeah, watch this space. The future's going to come.
We'll take one more question. Is there a question on the floor before we move on? No? Thank you very much.
Okay, thanks.